time is the enemy of evolution. Given enough time, the second law of thermodynamics says that things become more neutralized as time goes on. Things don't get better and better. You take a car, brand new car, drive it 1.6 miles from the car dealership to your driveway, then put a tarp over the car, never touch it again for 20 years. You never touch that car. At the end of 20 years, with a flourish, you pull away that tarp, and what do you have? A wreck. A wreck, and you've never driven it once.
You can walk for 1.6 miles, then put a tarp over yourself, and sit there for 20 years. What do you get? I tell you what you get, a wreck. We are not special, the second law is affecting us. But we do three things that the car does not. We breathe, we eat, and go to the bathroom. Every human spent about half an hour as a single cell. All life has to begin somewhere, and even the largest humans spent a short part of their lives as a single-celled organism when sperm and egg cells first combine. Shortly afterward, the cells begin rapidly dividing, and begin forming the components of a tiny embryo. 300 million cells die in the human body every minute. While that sounds like a lot, it's really just a small fraction of the cells that are in the human body. Estimates have placed the total number of cells in the body at 10 to 50 trillion, so you can afford to lose a few hundred million without a hitch. Humans shed about 600,000 particles of skin every hour. You may not think much about losing skin, if yours isn't dry or flaky, or peeling from a sunburn, but your skin is constantly renewing itself, and shedding dead cells. Every day an adult body produces 300 billion new cells. Your body not only needs energy to keep your organs up and running, but also to constantly repair and build new cells to form the building blocks of your body itself. Living cells and organisms must perform work to stay alive and to reproduce themselves. The synthetic reactions that occur within cells, like the synthetic processes in any factory, require the input of energy. Without energy, structures rich in information inevitably become disordered and meaningless. In the course of evolution, cells have developed highly efficient mechanisms for coupling the energy obtained from sunlight or fuels to the many energy-consuming processes they must carry out. We can consider cellular energy conversions like all other energy conversions in the context of the laws of thermodynamics. Living organisms exist in a dynamic steady state, never at equilibrium with their surroundings. The molecules and ions contained within a living organism differ in kind and in concentration from those in the organism's surroundings. A paramecium in a pond, a shark in the ocean, an erythrocyte in the human bloodstream, an apple tree in an orchard all are different in composition from their surroundings, and, once they have reached maturity, all maintain a constant composition in the face of constantly changing surroundings. Although the characteristic composition of an organism changes little through time, the population of molecules within the organism is far from static. Molecules are continuously synthesized and then broken down in chemical reactions that involve a constant flux of mass and energy through the system. The hemoglobin molecules carrying oxygen from your lungs to your brain at this moment were synthesized within the past month. By next month they will have been degraded and entirely replaced by new hemoglobin molecules. The glucose you ingested with your most recent meal is now circulating in your bloodstream. Before the day is over, these particular glucose molecules will have been converted into something else, carbon dioxide or fat, perhaps, and will have been replaced with a fresh supply of glucose, so that your blood glucose concentration is more or less constant over the whole day. Maintaining this steady state requires the constant investment of energy. When the cell can no longer generate energy, it dies and begins to decay toward equilibrium with its surroundings. For a chemical reaction occurring in a solution, we can define a system as all the reactants, products, the solvent and the immediate atmosphere, everything within a defined region of space. The system and its surroundings together constitute the universe. If the system exchanges neither matter nor energy with its surroundings, it is said to be isolated. If the system exchanges energy, but not matter with its surroundings, it is a closed system. If it exchanges both energy and matter with its surroundings, it is an open system. A living organism is an open system. It exchanges both matter and energy with its surroundings. Living organisms derive energy from their surroundings in two ways. They take up chemical fuels such as food from the environment and extract energy by oxidizing them, or they absorb energy from sunlight. The first law of thermodynamics, developed from physics and chemistry, but fully valid for biological systems as well, describes the principle of the conservation of energy in any physical or chemical change. The total amount of energy in the universe remains constant, although the form of the energy may change. The flow of electrons provides energy for organisms. Nearly all living organisms derive their energy, directly or indirectly, from the radiant energy of sunlight, which arises from thermonuclear fusion reactions carried out in the sun. Creating and maintaining order requires work and energy. DNA, RNA, and proteins are informational macromolecules. In addition to using chemical energy to form the covalent bonds between the subunits in these polymers, the cell must invest energy to order the subunits in their correct sequence. It is extremely improbable that amino acids in a mixture would spontaneously condense into a single type of protein with a unique sequence. This would represent increased order in a population of molecules, but according to the second law of thermodynamics, the tendency in nature is toward ever greater disorder in the universe. The total entropy of the universe is continually increasing. To bring about the synthesis of macromolecules from their monomeric units, free energy must be supplied to the system. The randomness or disorder of the components of a chemical system is expressed as entropy. Any change in randomness of the system is expressed as entropy change delta S, which by convention has a positive value when randomness increases. Yet cell function depends largely on molecules, such as proteins and nucleic acids, for which the free energy of formation is positive. The molecules are less stable and more highly ordered than a mixture of their monomeric components. To carry out these thermodynamically unfavorable, energy-requiring, endergonic reactions, cells couple them to other reactions that liberate free energy, exergonic reactions, so that the overall process is exergonic. The sum of the free energy changes is negative. The usual source of free energy in coupled biological reactions is the energy released by hydrolysis of phosphonhydride bonds such as those in ATP. In thinking about energy coupling, it is useful to consider a simple mechanical example. An object at the top of an inclined plane has a certain amount of potential energy as a result of its elevation. It tends to slide down the plane, losing its potential energy of position as it approaches the ground. When an appropriate spring and pulley device couples a falling object to another, smaller object, the spontaneous downward motion of the larger can lift the smaller, accomplishing a certain amount of work. The amount of energy available to do work is the free energy change, delta G. This is always somewhat less than the theoretical amount of energy released, because some energy is dissipated as the heat of friction. How does this apply in chemical reactions? In closed systems, chemical reactions proceed spontaneously until equilibrium is reached. Each compound involved in a chemical reaction contains a certain amount of potential energy. In reactions that occur spontaneously, the products have less free energy than the reactants. Thus the reaction releases free energy, which is then available to do work. Such reactions are exergonic. The decline in free energy from reactants to products is expressed as a negative value. Endergonic reactions require an input of energy, and their delta G values are positive. As in mechanical processes, only part of the energy released in exergonic chemical reactions can be used to accomplish work. In living systems, some energy is dissipated as heat, or lost to increasing entropy. In living organisms, as in the mechanical example in an exergonic reaction, can be coupled to an endergonic reaction, to drive otherwise unfavorable reactions. The coupling of exergonic and endergonic reactions through a shared intermediate is absolutely central to the energy exchanges in living systems. Living cells are open systems, exchanging matter and energy with their surroundings, extracting and channeling energy, to maintain themselves in a dynamic steady state distant from equilibrium. Energy is obtained from sunlight or fuels, by converting the energy from electron flow into the chemical bonds of ATP. The tendency for a chemical reaction to proceed toward equilibrium can be expressed as the free energy change, delta G, which has two components: enthalpy change, delta H, and entropy change, delta S. When delta G of a reaction is negative, the reaction is exergonic, and tends to go toward completion. When delta G is positive, the reaction is endergonic, and tends to go in the reverse direction. When two reactions can be summed to yield a third reaction, the delta G for this overall reaction is the sum of the delta Gs of the two separate reactions. This provides a way to couple reactions. The conversion of ATP to inorganic phosphate and ADP is highly exergonic. That means a large negative delta G, and many endergonic cellular reactions are driven by coupling them, through a common intermediate, to this reaction. Most exergonic cellular reactions proceed at useful rates, only because enzymes are present to catalyze them. The catalytic activity of enzymes in cells is regulated. Metabolism is the sum of many interconnected reaction sequences that interconvert cellular metabolites. Each sequence is regulated so as to provide what the cell needs at a given time and to expend energy only when necessary. The energy source that drives all living things is the sun. The energy of the sun is captured through photosynthesis, which produces glucose, which contains that energy in chemical energy form. This chemical energy is then transferred to living cells by cellular respiration, which produces usable chemical energy in the form of adenosine triphosphate ATP. The organelles we call mitochondria are found in the cytoplasm of nearly all eukaryotic cells. Their most immediate function is to produce adenosine triphosphate ATP by systematically extracting energy from nutrient molecule substrates. ATP is the universal energy yielding commodity in cells, used by enzymes to perform a wide range of cellular functions. We cannot survive, even for a moment, without a sufficient supply of ATP. We refer to the process as aerobic metabolism, because it requires constant removal of excess electrons through the reduction of oxygen. The need for oxygen as an electron acceptor is the sole reason that we breathe air. In fact, the most immediate purpose of our respiratory and circulatory systems is to deliver oxygen to the tissues for use by mitochondria and to eliminate carbon dioxide. By the way, the term respiration is synonymous with the phrase cellular respiration. It refers to the reduction of oxygen to water. Simply inhaling and exhaling is simply breathing, not respiration.